is going to talk about proteomics approaches in infection research. And then Professor Hamdi Uysal is going to talk about proteomics approach in diagnosis of cancer. Then we will continue with Professor Maren Kökritz-Bilek. I'm sorry for my pronunciation. She's going to talk about the role of hypoxia in the innate immune response against infections. Then Yeliz Kaya is going to talk about para-oxanase and their usage in the diagnostic and prognostic marker in veterinary medicine. And then Yasemin Salgırlı Demirbaş is going to explain us how she was able to develop a COVID-19 detection dog. And then Özge Sızmaz is going to talk about yeast wall and gut health in broilers. And the fifth session is going to begin. And we will start with Dr. Mikolaj Adamek, and he is going to talk about skin barriers' importance in the carbs. And then Dr. Farah Gonul Aydın is going to talk about the importance of antibiotic drug resistance in fish pathogens from One Health perspective. So I would like to invite Gisa Gerold to open her microphone, and we can start our presentation.
All right, thank you very much. I hope you can hear me fine. Maybe you can briefly
Yes,
confirm.
we can hear you.
Okay, great. So let's see, I guess you can see my screen also.
We can see the screen, but if you could, yes, click on yes. And then could you make one slide, could you forward one slide so that we have to make sure the slides are perfect. Thank you.
Working fine. Okay, thank you very much. So thank you very much to the organizers for having me. I'm really glad to be part of this symposium today. Actually, I just joined the TIHO, the veterinary school here in Hanover last year, so I'm really happy that this initiative here exists. So today I will give you a glimpse of our proteomics research that we do in infection research, and in particular in virology. And to start off with, I'm showing you this image of the galaxy because on our planet we know that there are more viruses present than stars in the observable universe. So we're talking about numbers of 10 to the 21. So lots of viruses on this planet, and we know that a number of these viruses have the potential to cause disease, and many of those we may not even know to date. We know also that 60, or I should say rather 70% of all human diseases have their origin in animals. So they are so-called zoonotic pathogens. And we also know that animal pathogens account for huge loss in the food-producing industry. And that is, with regards to animal-related food-producing industry, 20% of losses. So also this is infectious disease is also a big economic problem, of course. And according to the One Health concept, not only animal and human health are tightly intertwined, but also the ecosystem health. And we've learned this the hard way in the past 1.5 years during the COVID-19 pandemic, that most likely interference of humans with ecosystems can lead to loss of habitats and therefore to the spread of diseases from wildlife to livestock, but also to humans. So these three sectors are tightly intertwined: human health, animal health, and ecosystem health. And we need to be prepared in the future for possibly newly upcoming emerging viruses. And this is really the topic of our research here at the Research Center for Emerging Infections and Zoonosis at the University of Veterinary Medicine in Hanover. So pandemic preparedness and response, in my view, has 4 main pillars. And today, in the interest of time, I will only talk about the last one that's shown here to the right, the therapies. We are actually, in terms of COVID-19, lagging behind with the development of antiviral therapies. Vaccines luckily became available rather fast. So how can we contribute to therapy development? And we also have to work on COVID-19, but today I will talk about another virus. the key is really basic research, because I would like to give you here an example of a human pathogen, the hepatitis C virus. I've worked on this virus for many years, and we could understand on a cell biological level in detail the whole process of infection with this virus. So we could delineate the protein-protein interactions of the virus at the plasma membrane, so the virus uses specific receptors, but also intracellularly, where the virus translates its genome and replicates its genome with the help of host factors, host proteins. And this is a big success story, because based on this fundamental research on how the virus interacts with human host cells, a very efficient therapy has been developed over the years. And to date, hepatitis C is a curable disease. Within 12 weeks, you can cure a patient and this therapy has no significant side effects. And this is why actually one of my mentors, Charlie Weiss, together with Harvey Alter and Michael Houghton, received a Nobel Prize last year for the discovery of hepatitis C, but also for all their fundamental work on how hepatitis C interacts with host cells that now led to the discovery of these efficient therapeutics. And I think the same can be true for animal diseases and also for newly emerging zoonotic diseases in humans. So the question is, can we rapidly identify virus host factors? And I would say yes. There are many different methods that you can use, but a very successful method in our view, and we are focusing on this, is proteomics, mass spectrometry-based proteomics. This technology dates back many years, so it was the principle of mass spectrometry was discovered in the late 19th century at the same time when the first virus was identified. Both fields, the virology field but also the proteomics field, have made great advancements, in particular in the last 10 to 20 years. So for mass spec proteomics, basically the high computing power that we have now contributed to an explosion of the field, basically. So we have so many options now to understand infectious diseases using this technology, and I will just give you a glimpse of this today. So this is a summary or a cartoon showing the results of our work on hepatitis C, where we could actually delineate the protein complex at the plasma membrane. You see in red the homodimer of the receptor of the virus. This is called CD81, and we could identify the other associated proteins that you see here. These are growth factor receptors and integrins, but also intracellular factors that interact with the virus receptor, and we could show that these accessory proteins are also required for infection. And potentially you can interfere with these proteins, of course, to develop antiviral therapies. So this CD81 receptor is shown here. You have an ectodomain on the top where the virus binds, and an intracellular domain that also binds lipids. And through our work, we could successfully identify proteins that interact with CD81. The proteomic analysis is typically shown in such a volcano plot, as you see here on the left. And this is work of Lisa Laswitz, a PhD student soon to graduate in my lab. You see in this volcano plot basically statistical significance on the y-axis, so the higher up, the more significant. And on the x-axis, you see the enrichment of a protein. So what we do a co-immunoprecipitation. So we pull on the protein of interest, in this case the virus receptor, and check which other proteins are associated. And you see here that, of course, we find the receptor shown in green, and a known molecule, the scavenger receptor B1, is our positive control, shown with the big black dot, and also a couple of other new proteins. And we analyzed these in detail and could show, and Lisa could show that two proteins, calpain 5, shown here in red on the right-hand side, and Cybl B, shown in blue, are important host factors of the virus, because if we knock them out by CRISPR-Cas9 technology, the infection and the infectivity with hepatitis C virus strongly drops. Now here at the veterinary school, we are applying this technology to other viruses, and I would just like to mention, if you think of why is she telling us something about a human tropic virus, well, there are related viruses, Hep C viruses in animals, and this is work by many colleagues, also by Paul Becher here at the veterinary school, who discovered these viruses also, for instance, with Christine Bechlein in ruminants, in cows and also in other animals. So maybe we can learn, based on this experience on hepatitis C virus, also about the protein-protein interactions for these related viruses in animals. But now I would like to come to the viruses that are dear to our heart now. These are arboviruses, arthropod-borne viruses, and there are several animal pathogens, but also zoonotic viruses that are of relevance. I'm naming a few here: African swine fever virus, blue tongue virus, rift valley fever virus, for which I will show you an example today, West Nile virus, and also the equine encephalitis viruses, which we work on. Now, for a virus that is related to rift valley fever virus, and that is a safety level 2 surrogate. It's called Yukonimi virus. We did a proteomics approach that was slightly different to the one I just showed you. Here we affinity enriched the viral glycoproteins, shown here in red, GN and GC. Again, it's the same volcanoplot, same principle, and you can see a strong enrichment of the viral proteins, not only GN and GC, but also other associated viral proteins, namely L and N, and a number of host proteins shown in green. Again, we analyzed these in detail by RNA interference and could show that one of these proteins, GBF1, which is a protein that's important for intracellular transport between Golgi and ER, is a host factor for these Bunya viruses. We could also show that a couple of related viruses need this protein, and we showed this by either RNA interference or using a specific inhibitor, shown here on the right. This inhibitor is called Golgi side A, or GCA. And with this chemical compound, we can strongly suppress virus infection. Now, I would like to show you at least one data slide on on coronaviruses. We also do a lot of work on SARS-CoV-2 that is currently being published. But at that time, we were already working on the common cold coronaviruses, namely human coronavirus 229E. And you see here on the left-hand side, the RNA interference experiment. So 2 independent siRNAs against GBF1 could suppress infection with this common cold coronavirus to a certain degree, 50% roughly. And when we use this inhibitor, Golgi side A, on the right-hand side, you see a dose-dependent reduction in coronavirus infectivity upon treatment with this inhibitor. So this GBF1 protein that we identified is actually important for several zoonotic viruses, but we also found viruses like adenoviruses, for instance, that are completely refractory to treatment with this inhibitor. So there is some specificity, but we believe that this protein is important for a couple of viruses. Okay, so this already brings me to the end as a take-home. We are all convinced that future pandemics require preparedness strategies, and I didn't talk about the other pillars of pandemic preparedness, but one pillar is, of course, the development of antiviral approaches. And I think proteomics can really elucidate fundamental aspects of virus infection, actually also of other pathogens, not only viruses, and thereby help identify targets for therapies. For many animal and zoonotic pathogens, in fact, these protein networks during infection are insufficiently described, in particular in animals. In humans, we get more and more information, and therefore this really demands investigation. The field of proteomics is huge, I have to say, and this is just a scheme on the right-hand side illustrating this. Today I mainly talked about the protein-protein interactions. But we can also use proteomics to identify protein substrates, to identify post-translational modifications, to look at presentation of antigens, and also look at innate sensing mechanisms, to define pathology. Proteomics is used in diagnostics also more and more, and we can do spatial proteomics, which again in pathology, for instance, is important. So there are many, many applications, and we hope to be able to endeavor into several of these directions. If you want to read more about this, I recently edited a special volume in Advances in Virus Research on proteomics approaches, and there you can find detailed information about what is possible to date in this field of research. So with this, I would like to thank my wonderful team here in Hanover, which you see on this picture. Today I showed data from Rebecca and Lisa mainly. And I also thank my team at Umeå University in Sweden, where I have a guest professorship, and all the funding sources. If you like what you've heard, you can follow us on Twitter at Gerold Lab. And I thank you very much for your attention, and I hope that you all stay safe during fall and winter. Oh, this is not working. Okay, I wanted to show you this little movie of how to stay safe, but I think I will skip this. And with this, I thank you very much for your attention, and I'm happy to take any questions.
Thank you, Professor Gisa. Are there any questions? I actually have one.
Sure, go ahead.
So you are working with the whole protein structure, not the epitopes or or small parts of proteins in the viruses, or right? And
yeah, this is a good point. Yes, in principle, yes. So what we do is typical peptide fingerprinting. So in the end, during the process of analysis, we digest the protein into peptides. But initially, we work with the whole protein. But but go ahead, ask with your question. Sorry.
No. The question was that. I mean,
okay. Yeah. So. So
yeah.
in the future, we would also like to do immunopeptidomics, so to look at peptides that are presented by antigen-presenting cells, and now proteomics has the power to identify these and also quantify protein on epitope presentation. We haven't done it yet, but this is a plan for the future.
Okay, thank you. Are there any questions? If not, then we are going to continue with Professor Hamdi. You can close your presentation. Thank you. Hamdi Hocam.
Okay, yes, one moment. Okay, can you hear my sound, please?
Yes.
Okay, good, thank you very much. Dear participants, I would like to greet all of you firstly, and then I will start this talk, but I have to indicate that I have been in TIHO Hannover twice. Sorry, in 2000 and 2005, I've been in the Institute of Biochemistry. I have visited the labs of the Professor Naim. So since then, it was in 2005, I have visited by an Erasmus program. So I am quite familiar to Hannover University of Veterinary Medicine. So I am happy that to see this symposiums. They are starting again after several years. Now I can start my talk. Okay. So today I'm going to talk about the role of proteomics in diagnosis of cancer. So firstly, I would like to give some general backgrounds about the proteomics. Then I will concentrate on our studies in our laboratory in Ankara. So the term proteomics and proteome coined by Mark Wilkins and his colleagues in 1994. The proteome is described as the set of proteins expressed in a specific cell, tissue, or organism at a certain time. Proteomics can be defined as the systematic analysis of proteins for their identity, quantity, and function. So proteomics is the global analysis of hundreds to thousands of proteins in cells or tissues simultaneously. Basically, the two-dimensional electrophoresis is used for the separation of proteins on a matrix. Also, after that, identification of separated proteins in a high throughput way by mass spectrometry, it is studied. So one of the core technology of proteomics is the two-dimensional electrophoresis. At the present, there is no other technique which is capable of resolving thousands of proteins in one separation procedure. So in a protein separation, we have a two-dimensional electrophoresis system. So in this system, the first dimension is the isoelectric focusing, and then second dimension is the polyacrylamide gel electrophoresis. So mass spectrometry also, it is an analytical technique that ionizes chemical species and sorts the ions based on their mass to charge to ratio. It is used to identify proteins in a mixture, especially MALDI-TOF. It's a good example for this mass spectrometry in proteomics studies. So proteomics technologies for the diagnosis of cancer. So in this, for this purpose, two-dimensional gel electrophoresis, mass spectrometry, and laser capture microdissection methods, they are very often used for the, as the proteomic technologies for the diagnosis of cancer. Actually, main target is the detection of the molecular markers of the cancer in this system. So when we say biomarkers, it's like cancer biomarker refers to that is indicative of the presence of cancer in the body. It is an important tool for cancer detection and monitoring. Proteins also, they are used as biomarkers like HER2 and all epidermal growth factor receptor. They are the examples. So proteomics is the opportunity for the detection of the biomarkers that provide the diagnostic and prognostic information about the pathogenesis of diseases. In recent years, the large number of proteomic studies to detect the biomarkers have been done. Comparative proteomic studies using two-dimensional gel electrophoresis coupled to mass spectrometry has been applied in various cancers, like including gastric cancer, lung cancer, prostate cancer, and breast cancer. Also, now I can talk about our studies in our laboratory. So we have studied the comparative proteomic analysis of fibrosarcoma and skin fibroblast cell lines. So in this study, different protein expressions between human skin fibroblast cell line and human fibrosarcoma cell line were studied by two-dimensional gel electrophoresis and liquid chromatography coupled with tandem mass spectrometry, which is LC-MS-MS techniques. So we compared the intracellular proteins by preparing lysate samples and the extracellular proteins by preparing the secretome samples between fibrosarcoma and fibroblast cell lines. The aim of the present study was to understanding the molecular mechanism of the fibrosarcoma and investigate the potential biomarkers for the fibrosarcoma. So we have established the human skin fibroblast cell lines and human fibrosarcoma cell lines, which were purchased from the ATCC, and they are established in our laboratory, and then I will not go to details of it. So also from the cell culture systems, we prepared the secretome samples, which contains the secreted proteins. Also, we used this as a sample in our experiments. Also, another preparation was the cell lysate samples. So we also prepared the cell lysate samples from the 70% confluence of the cells. So in the system, two-dimensional electrophoresis is the powerful technique for the separation of proteins. So after two-dimensional electrophoresis, gels were stained with the colloidal blue staining. So I am not going to talk about the details of this technique, but also stained gels after two-dimensional electrophoresis were scanned using densitometer and also analyzed by PDQS two-dimensional electrophoresis analysis software. Also, gels were treated with the trypsin digestion solution to get the polypeptides before the analysis in LC-MS. So for the LC-MS analysis, we applied this secretome and lysate samples then we have the results from this analysis. But peptide mass measurements were performed, and finally protein sequence database from the UniProt was used in our experiments. So also cell viability was determined by trypan blue exclusion method. Now I can concentrate on the results to talk about. Also, like this slide, for example, this representative images of two-dimensional electrophoresis maps of fibroblast cells, fibroblast lysates, and also fibrosarcoma lysate samples here. But differentially expressed proteins between human fibrosarcoma lysate and human skin fibroblast lysate samples, the numbered spots represent the proteins up and down regulated in fibrosarcoma lysate samples. So these are the numbered spots. They are the up and down regulated protein spots in this analysis. Also, this slide shows the differentially expressed proteins between human fibrosarcoma secretome and human skin fibroblast secretome samples. Also, the numbered spots represent the proteins only expressed in fibrosarcoma secretome samples. Here is the results of this especially differentially expressed proteins between two lysate groups of human fibrosarcoma and to human skin fibroblast cell lines. So we have observed about 14 proteins. They are differently expressed in this lysate groups of fibrosarcoma samples especially. Also, differentially expressed proteins between two secretome samples. Also, they are the these samples, they contain the extracellular proteins of human fibrosarcoma and also human skin fibroblast cell lines. So these are about 13 proteins we have observed. They are differentially expressed as the extracellular proteins, especially in human fibrosarcoma cell lines. So in this study, we identified 14 proteins in lysate samples and 13 proteins in secretome samples. In lysate samples, 13 identified proteins were overexpressed and 1 protein down expressed in fibrosarcoma. And in secretome samples, 13 identified proteins were only expressed in fibrosarcoma. Also, the heat shock proteins such as mortalin, endoplasmin, and heat shock protein 60 were found overexpressed in fibrosarcoma lysate samples. Also, TCP1, beta, and epsilon are molecular chaperones and were found overexpressed in fibrosarcoma lysate samples. Chireticulin is also molecular chaperones and was only expressed in fibrosarcoma secretome samples. Also, co-chaperone, stress-inducible protein 1, was found overexpressed in fibrosarcoma lysate samples. So among the upregulated proteins, heterogeneous nuclear ribonucleoproteins A2 and B1 and the MYC downstream regulated 1 proteins, they are increased in fibrosarcoma lysate samples. Also, it is known that heterogeneous nuclear ribonucleoproteins A2, and B1 is strongly associated in cell proliferation and cell survival in cancer. Also, this NDRG1 has potential function such as cell differentiation and cell regulation. Also, this protein was increased in the other cancers like gastric cancer, lung cancer, breast cancer, and adenocortical carcinomas. So this protein was overexpressed in these cancers also. Also, galectin-1 sparge urokinase type plasminogen activator and isoform APP751 of amyloid beta A4 proteins were secreted proteins, and compared to fibroblast secretome, these proteins were only detected in fibrosarcoma secretome samples. So we detected a remarkable down expression of the major Volt protein in fibrosarcoma cell line. Among the 27 identified proteins in lysate and secretome samples, 6 metabolic enzymes such as protein disulfide, isomerase A3 and A6, alpha-enolase and peptidyl prolyl cis-trans isomerase A and B were identified. These enzymes have involved many biological processes such as protein folding and glucose metabolism. Proteomic separation and analytical techniques are uniquely capable of detecting tumor-specific alterations in proteins. The comparative proteomics studies are essential to understanding the molecular mechanisms of the cancer and biomarker discovery. Overall, our results will benefit the identification of potential tumor markers and understanding of the mechanisms of the fibrosarcoma. So first, I would like to thank the Ankara University Research Projects Office for supporting our studies. Also, I would like to thank my colleague Övünç Meral, who is a great proteomics person especially for two-dimensional electrophoresis. He did great study, and thank you very much to him. I appreciate very much. Also, this is the bird's eye view of our little campus in Dışkapı in Ankara. So this is the white building. We are, as biochemistry department, we are at top of this building. So this is our little campus for your view. So thank you very much for your attention. I'm sorry I a little went fast, and I hope you are okay.
Yes, it was longer than expected, but it's okay. The topic was very interesting. And we have a question from Gisa. Yeah, please, you can ask.
Yeah, thank you very much for this interesting insight. I was wondering, so I guess after you do the GL analysis, you do data-independent acquisition mode, right? When you measure the tryptic peptides?
So I'm sorry, I couldn't catch your sound.
Sorry, I hope you can hear me fine. I was just wondering, in the initial identification of putative biomarkers, did you do data-independent acquisition mode for the MS analysis? And later on, probably data-dependent, or can you select?
No, we just, LC-MS analysis, we analyzed these lysate samples and secretome samples, but from this analysis, I only pointed out the overexpressed and especially overexpressed proteins particularly. So these are the selected, especially selected ones in fibrosarcoma samples.
Okay, great. And then I assume that when you do the analysis from tissue samples, you don't run 2D gel electrophoresis anymore, right? So you do this directly from mice.
I know, because these times depends on your approach. I know you apply the global, mixtures to the system. Then you get the samples. Because proteomic science is every day is changing. Maybe you may see the necessary, depends. But you see, in our laboratory, we have, I can say, we don't have all the techniques maybe you have in Germany, but we have the two-dimensional system that we can use it. But of course, if we wish to get mass spectrometry like MALDI-TOF or LC-MS, these are, and also every day new techniques are coming out, but also they are the expensive,
yes,
systems to
I know.
get it. But I understand, I appreciate your, approach. Thank you very much.
I've done 2D gel electrophoresis before. It's a very valid approach, and so the data looks very solid. Thank you very much. Yeah.
Thank you. And also nice to meet you, Professor Gerold, because as you know, twice I've been in your institute. Since then, you have two professors, one of you, and also another speaker just coming after me. And so I'm very pleased to meet you here also.
Yeah, I can say exactly the same. So pleased to meet you, and hopefully at some point in person then. Okay,
I hope we keep in touch from now on.
Okay, so I am sorry to interrupt. We will continue with Maren von Kökritz-Blikwede, and I hope my present pronunciation is good now.
Yes, perfect. Thanks a lot.
So we are, yeah, you can start your talk. Do you need any help with sharing the presentation, or you are good?
Yeah, it should be fine. I hope it is appearing soon. Do you see my presentation already and my video?
We are not seeing it yet because it takes several seconds for us to catch it. Now we see a black screen.
Okay.
Yes. No, you are now sharing your main screen. You need to pick the slide and click on it and say share. Let's see.
I thought I did that.
Okay.
You have to open your PowerPoint, Maren. We can see your screen, but not your PowerPoint.
Okay, let me try it again.
Just click on the PowerPoint presentation if it's open, and then on the full screen mode there. I think then it should work. If you go on share application or share screen.
Now it works, right?
It takes a second. Yeah, super.
Do you see the full screen mode?
No, now we see slide 2, and now you have to go on the full screen. Yes, slide 1, but not.
And perfect, yes.
Do you see the full screen now?
Yes, we see the first screen now, and
also in the full screen.
No,
not yet.
We cannot. Is it in the presentation mode? No, right?
Können Sie nochmal drücken, diese Fullscreen?
In the worst case, I could try to share, but we see your presentation, so you just have to go into the full screen mode, I guess.
Yeah, I have been in the full screen mode. I don't know why it's not working.
Okay.
Let me try it once more again.
Yeah, perfect. Perfect.
Full screen mode?
Yeah.
Okay. And okay, it's a great pleasure now to present our ongoing work in the research group Infection Biochemistry, also at the Department of Biochemistry at the TIHO. And our work focuses on the role of hypoxia in the innate immune response against infections. So now I switch to the second slide. I hope that worked. Can somebody please quickly confirm?
Yes, it works.
Perfect. So I think, as we all agree, the understanding of the host-pathogen interaction is the basis of finding new therapeutic strategies. However, what I want to highlight today is that most of our in vitro experiments when studying host-pathogen interactions are performed under atmospheric oxygen conditions in the normal tissue incubator at 21% or a bit lower when we have CO2 at 19%. But in the healthy host, the physiological oxygen content in the tissue, depending on the tissue where it's localized, is below 21%. And especially during infection and inflammation, when immune cells infiltrate and also, for example, pathogens consume oxygen, then the oxygen level significantly drops down to hypoxic conditions. And hypoxia, we know from the literature, affects the response of host cells, and also pathogens adapt to their metabolism to hypoxia. And in order to find new treatment strategies, we need to mimic in vitro the physiological or also pathophysiological oxygen levels that occur during an infection. And this is what we study in our research group. We currently have a BMBF project which is called Hypoxia Infect. And there we study the role of hypoxia in Streptococcus suis infections. We have heard already from Professor Peter Valentin-Weigand about Streptococcus suis as a zoonotic pathogen, and it is able to cause severe problems in human and also in pigs by leading to meningitis. So the bacteria infiltrate via the choroid plexus epithelium to the brain and especially are able to survive in the cerebrospinal fluid and lead to meningitis. And a typical phenomenon during the innate immune response is that high amount of neutrophilic granulocytes are infiltrating and modulating the immune response and also have an impact on the infection in human as well as pigs. So our goal is to study the role of hypoxia in this infection. So as I mentioned, phagocytes, especially the neutrophilic granulocytes, play a key role. And we all know that phagocytes have different roles when encountering a pathogen. One role is phagocytosis, the intracellular killing, then also cytokine release or degranulating antimicrobial components. However, in 2004, a quite interesting phenomenon was described that especially neutrophils, but also other phagocytes have the ability to release extracellular DNA and to use this DNA to entrap and kill microbes. This was published in 2004 by a group from the Max Planck Institute in Berlin, Volker Brinkmann and Arturo Zichlinski, and they published this phenomenon in Science and discovered that neutrophils release their DNA to the environment, and in the environment, nets are formed which are able to then entrap pathogens and mediate an inflammatory response and also being protective against certain bacterial pathogens. And we are asking the question in my lab, so how can a phagocyte decide to either perform phagocytosis or also, on the other hand, to release these extracellular traps, or short called nets? And we think that the oxygen level in the tissue or in the infectious process plays one of the key roles in manipulating or in modulating neutrophil responses. So during an infectious process, neutrophils infiltrate to the tissue, then also they attract other immune cells. All these immune cells consume oxygens besides pathogens, and this leads to drop of oxygen level, hypoxia. And this finally influences the cellular behavior and host pathogen interactions. And in the lab, we try to mimic this situation by using 3D cultures of epithelial cells, in this case, choroid plexus epithelial cells, to mimic the human blood or also pig blood CSF barrier. And we adjust this model to hypoxic conditions. So how does this model look like? We cultivate epithelial cells which are polarized on transfer filters. We also infect them with bacteria like Streptococcus suis. We add blood-derived neutrophils to the system that are able to transmigrate through the epithelium, and then we can study this interaction in a transfer system. However, when we want to apply oxygen conditions to the system, the key question is arising: what are actually the physiological oxygen levels in the infected tissue? When we consider oxygen level, we have to talk about three terms. One is the normoxia, the normal atmospheric condition that we also find in the tissue incubator, and physioxia, which is the physiological oxygen level, and hypoxia, what is found under inflammation and infection based on a drop of oxygen in the tissue. When we look in the literature, there are actually information found on physiological oxygen level, for example, in the brain, which lies around 4.4%. which you see here in the table in a review that was published 2011 by Caro et al. However, no data are available about how are oxygen levels, especially during infection, and what are pathophysiological oxygen level. So the problem is there are no data available in vivo, especially not in the cerebrospinal fluids. And that was our goal in one of the studies, that we want to characterize on one hand the oxygen level in our current in vitro system, and also measure them in vivo in living pigs and also in infected pigs, compare these data that we get in vivo and in vitro, and adapt our in vitro models to the in vivo relevant situation to run then our infection studies under real closely mimicking in vivo situation, oxygen and also pH conditions. So we started first with a measurement system that we established in the lab. It's a non-invasive fluorescence-based oxygen measurement system to characterize the oxygen levels in our in vitro system. So we added an oxygen-sensitive sensor spot to our transval system that you see here with the red circle. And then in the transval system, we can measure oxygen levels in just the plain cell culture with epithelial cells, but also during infection with Streptococcus suis, and also when neutrophils are transmigrating in the system, if this changes the oxygen and pH situation. Then in the second step, we performed an animal experiment with infected piglets, and we infected the piglets with Streptococcus suis, the serotype 2, strain 10. And then the piglets developed meningitis. Then for ethical reasons, we put them under anesthesia, and a long time isoflurane anesthesia under controlled blood oxygenation. And then at several time points, we collect the cerebrospinal fluid and measure oxygen as well as pH, the pressure, as well as the colony-forming units of bacteria that we recover, and the amount of infiltrating granulocytes to the cerebrospinal fluid, and then perform histopathological analysis at the end. We compared also our data with animals that were euthanized to see how much there is the difference. And what we can clearly say is when we euthanize the animals, immediately after a few minutes, the oxygen drops to an unphysiological level, so which really explains the reason why we then performed this in vivo experiment under anesthesia to be able to measure physiological relevant oxygen levels. So we localized then a catheter so we in principle performed a cisternal puncture at the biggest subarachnoidal cistern, and then took the CSF and implemented a measurement system to be able to measure pressure as well as oxygen and pH under anesthesia that you can see here in the images. And so we had a big, let's say, experimental setup running to keep also the animals under the best conditions during the experiment. And then we show you now here the results that we got in vitro as well as in vivo. So on the left-hand side, you see our in vitro results from the transval system, and on the right-hand side, the results from in vivo experiments. So let's first focus on the left-hand results. So just the cells alone, the plexus epithelial cells alone, have an oxygen level of around 7%. And when later Streptococcus suis is infecting those cells, then the oxygen levels drop as we would expect. Also, when we add PMNs, so polymorph nuclear cells, so the granulocytes, then also the oxygen drops slightly. And even in combination, infected cells and transmigrating neutrophils, the oxygen drops even more. When we compare this with our in vivo data, we can see, oh, in vivo, in the cerebrospinal fluid, we see actually also oxygen levels of around 7% cerebrospinal fluid. The question was, how does that differ between infected and non-infected animals? You see here on the right-hand graph, the control animals which were not infected in blue, the infected in black and gray. However, we differentiated between infected no bacteria in CSF and in gray infected bacteria in CSF. However, what you see, at least during the course of our experiment, which mimics the early phase of meningitis, there was not a drop found in vivo during the experiment. So apparently the body was able to compensate the oxygen level and to keep it stable of around 7% in the CSF, also during infection in this early phase. So which was quite nice and interesting that our in vitro model with the plexus epithelial cells on the transfer filter, they mimic the situation as it occurs in vivo with 7%, at least with the plexus epithelial cells alone. However, after a long time of infection, here in this case 2 hours already, the oxygen drops depending on the amount of bacteria that are present in the system and which for sure can be modulated depending on the need also of the experimental design. However, what is important for us is that we are able with this method to generate in vivo relevant physiological data and adapt these data to our in vitro system and thereby contribute also to the 3R, to the reduction and replacement of animal experiments by optimizing our in vitro models. And that is our overall goal in this project. So here I want to thank all our cooperation partners, especially the key team in my research group, which are highlighted here in red, Nicole de Boer, Marita Meurer, and Alexander Martins, who worked on that project, and also our key cooperation partners from the small animal clinic, Professor Sabine Kästner, who helped us with the anesthesia of the animals, Christoph Baums from Leipzig, and also Professor Schroten and Professor Schwerk from Mannheim. Yeah, thanks for your attention.
Thank you. Are there any questions for Professor Maren? I guess not. Then we are moving on to the next speaker.
Okay, thanks a lot.
Thank you very much. Yeliz Kaya. Okay. Can you share your presentation?
Yes. My title is not okay. Okay.
Hi.
Hello. Can you see the image?
Now we can see, yes.
Okay. Guten Tag. Heute werde ich meine Präsentation in Deutsch vorstellen und willkommen zu dem Kooperationssymposium 8. Ankara-Hannover Kooperationssymposium und ich hoffe, wir können nächstes Jahr in einer gesünderen Welt zusammenkommen und nun fange ich an, meine Präsentation zu vorstellen. Heute ist meine Präsentation über Paroxanase und Paroxanase, also PON, ist ein Enzym, das erstmals in toxikologischen Forschungen entdeckt wurde und ist ein Glykoprotein mit antiaterogenen und antioxidativen Eigenschaften und wird stark an HDL gebunden und ist auch ein gemischtes Enzym, das verschiedene Substrate hydrolysieren kann. Paroxanase spielt eine wichtige Rolle beim Schutz vor Oxidation und bietet Schutz vor Lipidperoxidation und ist eine calciumabhängige Esterhydrolase. PON-Proteine sind bei Säugetieren weit verbreitet, aber in Fischen, Vögeln und wirbellosen Tieren sowie Arthropoden fehlen diese Proteine und dieses Bild ist die 3D-Struktur der Paroxanase. Bei Menschen und Mäusen liegen 3 separate PON-Gene nebeneinander auf demselben Chromosom und sie werden als Paroxanase 1, 2 und 3 ernannt. Es ist bekannt, dass das in der Leber synthetisiert und an den Kreislauf abgegebene PON1 in der HDL-Struktur stattfindet. Die Hauptsynthese von PON1 und PON3 findet in der Leber statt. Nach der Synthese wird ein Teil der Enzyme durch das spezifische Transportsystem von der Leber ins Plasma übertragen. Seine Verbindung mit Lipoproteinen wird im Plasma bereitgestellt. HDL ist das wichtigste Lipoprotein, das mit PONs im Plasma verbunden wird, aber in PON 2. Es wird nicht mit HDL assoziiert und ist ein intrazelluläres Protein. Eine hohe Expression außerhalb der Leber findet jeweils in Lunge, Plazenta, Hoden und Herz statt. Es wird vermutet, dass PON 2 protektiv ist, da es die Akkumulation von Superoxidanionen bei intrazellulärem oxidativen Stress verhindert. Es wird angenommen, dass PON 1 von dem gezeigt wurde, dass es in den Epithelzellen der proximalen Tubuli lokalisiert ist, einen funktionellen Beitrag zur Entgiftung von Xenobiotika leisten kann. Die bekannteste Funktion ist und besteht darin, aus Organophosphaten gewonnene Nervengase und Insektizide durch Hydrolyse unschädlich zu machen. Und es bietet auch Schutz vor Toxizität von bakteriellen Endotoxinen. Und es hat die Fähigkeit, langkettige oxidierte Phospholipide zu hydrolysieren. Ferretti und Mitarbeiter untersuchten Lipoprotein, oxidativen Stress und HDL-PON-Aktivität bei gesunden und adipösen Personen und zeigten, dass die HDL-PON-Aktivität bei Adipösen im Vergleich zur Kontrollgruppe signifikant niedriger war, und sie schlugen vor, dass die Veränderungen des HDL-Gehalts die Enzymaktivität verringerten, indem sie die Bindung von Paroxanase an die HDL-Oberfläche beeinflussten. Außerhalb der Fettleibigkeit, koronare Herzkrankheiten, Leberkrankheiten und neurologische Erkrankungen sind auch sehr wichtig, und Paroxanase 1 ist hier auch eine sehr wichtige Biomarker. Aber das Wichtigste, eine der wichtigsten ist Paroxanase und Krebs. Oxidativer Stress ist einer der wichtigsten etiologischen Faktoren bei der Karzinogenese. Da PON 1 andererseits zu den endogenen Systemen zum Abfangen freier Radikale gehört, ist seine Rolle bei der Etiologie und Prävention verschiedener Krebsarten von Interesse. Es wurde festgestellt, dass die PON-1-Aktivität bei gastroösophagealem Krebs und ähnlich signifikanten Entzündungen und krebsbedingter Anämie verringert ist. Und eine Verringerung von PON-1 hat sich als Hinweis auf Lymphknotenmetastasen gezeigt. Und nun kommen wir zu den Studien, die in unserer Abteilung gemacht wurden. Und dies ist die erste Studie, und hier sehen wir die PON-1-Aktivität der ovarioektomierte und ovariohysterektomierte Hunde vor und nach der Operation. Und wir können sagen, um die Schärfe der postoperativen Akutphasenreaktion zu reduzieren, ovarioektomierte Hunde sind übergeordnet als die ovariohysterektomierte Hunde, und PON-1-Aktivität des Hundes verändert sich nicht in den beiden Gruppen für die postoperative 4. Tag. In der zweiten Studie diese Studie ist über die PON-1-Aktivität der türkischen Sahnenziegen vor, an und nach der Geburt. Als Ergebnis kann man sagen, dass das höchste Aktivität, also PON-Aktivität, an der 30. Tag vor der Geburt beobachtet wurde, und an der Geburt, also an dem Geburtstag, weil die oxidativer Stress Maximum ist, wurde die PON-1-Aktivität viel niedriger gemessen. Dies ist die dritte Studie, und das Ziel dieser Studie ist es, die Veränderung der PON-1-Aktivität bei diabetischen und nicht-diabetischen Ratten zu untersuchen, denen Maulbeerextrakt verabreicht wurde. Die Gruppenbildung bestand die Kontrollgruppe aus gesunden Tieren, und denen wurden für 56 Tage lang nur Trinkwasser gegeben. In der ersten Gruppe da sind diabetische Ratten, und es wurde ihnen auch nur Trinkwasser angegeben. Und in der zweiten Gruppe, da sind wieder gesunde Ratten, und es wurde ihnen Maulbeerextrakt verabreicht für 56 Tage lang wieder. Und in der dritten Gruppe, da sind wieder die diabetische Ratten, und es wurde ihnen auch Maulbeerextrakte gegeben. Die Unterschiede zwischen PON-1-Aktivitäten erwiesen sich als statistisch signifikant. Die höchste PON1-Aktivität wurde in der Kontrollgruppe und die niedrigste in der dritten Gruppe beobachtet. Die Tatsache, dass das niedrigste PON in der dritten Gruppe lag, kann darauf zurückzuführen sein, dass Maulbeerextrakt die Verwendung des PON-Enzyms erhöht. Wenn man dies betrachtet, kann man sagen, dass die antioxidativen Eigenschaften von Maulbeerextrakt begrenzt sind. Und hier ist die vierte Studie wieder gemacht in unserer Abteilung. Diese Studie wurde mündlich auf einem internationalen Kongress vorgestellt und zielte darauf ab, den Einfluss des Alters auf die PON1-Aktivität zu untersuchen. Und hier können wir sehen, dass die niedrigste PON1-Aktivität in der ersten und zweiten Gruppe sind. Diese zwei Gruppen sind Ziegen bis zum den ersten Lebensjahr. Aber die höchste PON1-Aktivität können wir in der dritten Gruppe, also in der jüngeren Gruppe sehen. Dann die Aktivität von PON1 mit zunehmendem Alter wird verringert. Diese geringe Aktivität in den ersten zwei Gruppen kann auf das noch nicht voll entwickelte postnatale Immunsystem zurückzuführen sein. Und es gibt noch zwei weitere aber unveröffentlichte Studien in unserer Abteilung. Und die erste davon bezieht sich auf die Veränderung der PON zwischen Kulturrassen und Hauskatzen. Und da waren auch einige Unterschiede zwischen den PON-Aktivitäten. Da haben wir noch einige Literaturen, die Veränderungen in Schwangerschaft. In der Studie von Schimschek und den Mitarbeitern wurde festgestellt, dass die PON1-Aktivitäten von Schwangeren höher sind als die von Nichtschwangeren. Und da sind auch Veränderungen in der Rassenunterschiede und in Demirs Masterthese wurden die PON1-Aktivitäten von Jersey, Simmental und Holstein-Rindern verglichen. Es wurde festgestellt, dass die PON1-Aktivitäten in Jersey und Simmental-Rindern höher als in Holstein-Rindern waren. Und die Veränderungen im Alter in der Studie von Ceresa wurde bei mittleren Alters und bei Älteren eine deutliche Senkung der PON1-Aktivität beobachtet. So beobachteten wir in unserer Studie das auch. Und während PON1 ein klinisch wichtiger Biomarker bei Krankheiten ist, ist es auch ein wichtiger Biomarker bei physiologischen Ereignissen sowie Alter, Schwangerschaft und Entwicklungsstadien und sogar Rassen. PON1 könnte eine Rolle bei der Bekämpfung von oxidativen Stress bei Schwangeren spielen und bei Simmental und Jersey-Rindern, die Grund dafür wird angenommen, dass die Anpassung der Simmental und Jersey besser ist als die der Holstein-Rinder. Danke, dass ihr mir zugehört habt und danke für eure Geduld.
Thank you, Yeliz, for your presentation. Do we have any questions for Yeliz? I guess not.
How can I end this slide?
Sorry?
How can I end this slide? Okay,
Yeah.
it's okay. Thank you.
You're welcome. So we are moving on to our next speaker, Associate Professor Dr. Yasemin Salgırlı Demirbaş. And she's going to talk about COVID-19 detection dogs.
Thank you, Dr. Bilgen. Let me share my presentation first. Now, can you see it?
We can see you now.
You can see me?
And your presentation.
No?
It takes, yes. And you can switch to the presentation mode so we can continue.
It's already on the presentation mode, actually.
Okay, it takes several seconds for us to catch with your screen. Catch up.
Full screen yapmak gerekiyor.
Hocam şu anda full screen'de aslında. Ama bir daha çıkıp isterseniz yapın.
Yani görünmüyor öyle, tek tek görünüyor.
Now?
Sadly, no.
Okay, then maybe it's better to share the whole screen like this.
And share again.
Can you see it now?
Okay.
Okay.
Now we see that you are sharing the content. Yes, now it's okay.
Okay, and I'm moving forward. Can you
Yes,
see my screen?
yes.
Okay, great. So thank you again and good afternoon, everyone. Before starting my presentation, I would like to say that it's for me a great pleasure to present our project at this symposium as a former doctorate graduate of the Hannover Veteriner University. I have beautiful memories of Hannover, and Hannover has a very special place in my heart. So today I'm going to present our ongoing project on COVID-19 detection dogs. And first, I would like to give a brief overview of the background of this project idea. This ongoing COVID-19 pandemic showed us that preliminary screening of asymptomatic patients and pre-symptomatic patients in crowded areas such as airports or concerts is very important to reduce the risk for spreading infection. Of course, there are some methods we are currently using in such places for this aim, such as infrared thermal image scanners and rapid antigen tests, but none of these methods are sensitive enough to detect these asymptomatic patients and again pre-symptomatic patients. So since the beginning of this pandemic, authorities have emphasized that we need to develop new diagnostic methods which can be used in the field. And these diagnostic methods should be sensitive, fast, accurate, economical, and mobile in order to be used in such places, in everywhere. I mean, in schools, in airports, in all kinds of scenarios. And among these detection tools, actually, medical detection dogs came to the fore because they meet all these criteria. Actually, these dogs have been used for several years in different countries for diagnosing different diseases, and we know that they are capable of detecting volatile organic compounds which are released from the cells in case of any metabolic changes, and they are specific to the infection or the disease. And these dogs can detect this volatile organic compounds close to the femtomolar level. So their diagnostic accuracy is and sensitivity also are very high. They are higher than those of the routine methods. So United Kingdom was the first country who announced that, or which announced that they started training their COVID-19 detection dogs. And the team leader claimed that if they could train their dogs, these dogs could sniff up to 250 people per hour in public areas. And they were planning to use these dogs mostly in airports. And subsequently, a great deal of research has been published on medical detection, or on COVID-19 detection dogs, actually. And all of these studies show that these dogs can detect COVID-19 with greater sensitivity and specificity than routine diagnostic methods. So these results were very promising. But together with these researchers and with these results, some ethical and technical concerns have also emerged. For example, the big concern was that these dogs, or this was the first time that these dogs would be trained for detecting an infectious disease, which is highly contagious and has zoonotic potential. And interestingly, only a few studies mentioned these safety procedures during training, and none of these safety procedures can be applicable to the field. And these recent events with minks, I think you also heard about it. In Denmark, millions of minks have been killed because of the transmission between animals and humans, and this execution leads to highly controversial discussions which cover the public health and animal rights perspectives. So we should be very careful if we want to conduct these kind of studies in order to protect the health of public and also the health of dogs and the animals or the well-being of animals. And since the beginning of all these discussions, we, our research team, have followed all these discussions and publications. And our first priority actually was to establish a safe and applicable methodology for training these dogs as COVID-19 detection dogs. And we, of course, we wanted to use these dogs in the field, but we have to ensure the health of the dogs and the people who are in contact with these dogs. And these were going to become our first medical detection dogs in Turkey. So we had no experience on medical detection dogs. Only our dog trainer, as you see here, had some experience. But as I mentioned, this was our first experience with medical detection dogs. And another phase of the study was including the chemical phase. So we wanted to identify the disease-specific volatile organic compounds by analyzing these samples. So we had two important questions in the beginning. So one of these questions was, which sample should we use in the training and also in the field? And we set some criteria for sampling. It should be easily collectible because our medical team was working in a very harsh conditions. So we didn't want to put an extra burden on them. And also these samples should be used in the field as well. So after discussion, several discussions with the team, we decided to use face masks because we thought that face masks could be good candidate as they includes volatile organic compounds specific to respiratory infection, and it's a new normal for all of us. Everybody's wearing face masks, and it's a non-invasive method. And our second question was which decontamination procedure we should conduct. And we wanted to test different decontamination procedures. This decontamination procedure should be effective. We should inactivate the virus, but also in the field without changing the odor. Sorry. So we decided to test different decontamination methods. In our study, we trained 3 dogs. In total, we started with Zippo's training, and then in the second phase of this study, I will explain this just in a second. We will train another 2 dogs, Ruffle and Furfur. This is Zippo, and the black one is Ruffle, and English Cocker is the Furfur. And we had only 2 inclusion criteria, trainability and healthy. Actually, 2 of our dogs had no previous history of scent work at all. And Furfur, for example, this English Cocker Spaniel, had no previous history of training. So after we received or we got our ethical approval, we started our project. We divided our methodology into 2 phases, as I mentioned before. Methodological development phase, in which we wanted to test whether inactivated masks can be used in training for these dogs. And we conducted 2 different decontamination procedures. Hold-on procedure was a safe procedure. We were sure that we would not affect the odor of the sample, and we were sure that we activated the virus. And we wanted to test the heat procedure, the effect of heat procedure as well. And we used the masks of patients belong to different groups, and we also tested this effect, whether the different samples affected the performance of the dogs. And after we set our methodology and after we were sure about our methodology, we started the second phase of this study, application to field phase. So we trained another 2 dogs by using the same methodology, and we tested these dogs in and outer environment with or without humans, or in crowded areas. And for this step, we also tested another decontamination procedure in which we use UV light inactivation with a simple machine, as you may see here. So this, I hope you can see it. This is our dog, Breves. And here, this is the second phase of the study, and we are using an aluminium sand platform. And here there are 3 negative samples and 1 positive sample. And she is marking the sample by sitting. And because the owner had no idea which one is positive, he is asking whether it's correct. And after he got his approval, he rewarded the dog. And as you may see here, the diagnostic accuracy rates, sensitivity rates, specificity rates were similarly high of these dogs. And now we are conducting the chemical analysis phase of this study. We are using Headspace GC in order to analyze the volatile organic compounds. We had a chemical team, researchers from the chemical departments, and they are conducting this part of the study. So as a conclusion, we have seen that these dogs can be trained to detect COVID-19 by smelling the contaminated face masks with a significant success rate. And Holton and UV light procedures can be applied to inactivate these samples for the training of these dogs. And UV light inactivation has further a good potential to be used as a fast inactivation method in the field. So this project was supported by Turkish Scientific and Technical Research Council, and I'm thankful to our supervisors, Professor Dr. Aykut Özkul and Professor Dr. Gonçalo de Graça Pereira, and I would like to express my gratitude to our research team for their valuable inputs to this project. Thank you very much for listening.
Thank you very much for your presentation, Yasemin. Are there any questions for Yasemin? That's good. And yes, Professor Çiğdem, you can ask your question.
I have a question. Very nice presentation. For this presentation, I would like to thanks, first of all. I have a question about infection. We know that dogs can infected with SARS coronavirus too. What is the risk factor for this situation? What do you mean?
You mean during training or during using these dogs? In our project, actually, we didn't use sample directly for training. We first inactivated or decontaminated the masks by using hold-on procedure, in which we waited for a period until we were sure that all the viruses were inactivated, or we used heat procedure to inactivate these viruses. So we didn't want to put any risk to your dogs or to our trainers. We never used samples directly. We always conducted a safety procedure before the training and before the application step.
Okay, if I use this method, actually, generally, normally, naturally, and if the dogs can infect with this virus, it is high risk for this infection, I think.
Yeah.
What do you think about this?
It was actually, it is one of the biggest concerns about these dogs, because as you mentioned, these dogs are smelling the, not infection, actually the infection-specific volatile organic compounds. And if you use them directly, if they smell these samples directly, of course, this virus can be transmitted to dogs, and this maybe has a zoonotic potential, so this may transfer to the people, and it's a big health issue for the public. So as I mentioned in my presentation, this Minsk execution is a big lesson for everybody. Actually, it's a very sad lesson, but if you make these animals and humans in contact with each other in this kind of situation, it's a big risk. So we didn't want to take this risk, and I never suggest to use the dogs directly for this aim.
Okay, thank you very much.
You're welcome.
Thank you again. And we have also a question from the chat room. Apparently, Zoetis produced a COVID-19 vaccine for the zoo animals. And primates, panthers, skunks, and otters are prioritized, but maybe soon we'll have also specialized vaccines for the dogs. Is it possible? And also, do you think it can be also used for the scamp dogs in near future, this kind of vaccine, in order to protect the dog and also prevent it to maybe spreading the disease, something like that? Thank you, question for the study.
It would be great if we can vaccinate these dogs. It would be great, but if we want to use these dogs for another infections, because we know that the new infections are coming, we have to find out the safety procedures in any case.
Okay, thank you. I couldn't catch the last of your sentence. And are there any questions? Thank you again. And we are moving on to our next speaker. Associate Professor Özge Sızmaz is going to talk about yeast cell wall and gut health in broilers. Professor Özge.
Now, you hear me? Okay, thank you. Welcome to my presentation. Hello, dear all participants. Thank you for the organization committee for this opportunity to talk about here and also contact with Hannover. Yes. Today I'm gonna, I'm going to talk about the gut health in broilers and also the yeast and yeast cell components. It's well known that the gut health is fast becoming a reality in the monogastric animal industries. This is because the maintenance and of course the improvement of gut health is important for the welfare and the productivity of animals when antibiotics are not allowed in feed. Today, gut health is a major topic, not only in humans, but also in animals, of course. It's now generally considered that maintenance or enhancement of gut health is far more complex than just the modulation of the gut's microflora through some feed additives, such as probiotics, prebiotics, and also histomorphology, fermentation parameters, gene expression is seriously studied about that. The microbiota, gut morphology, and immune systems works together in the gut for the gut health. And also there is a relationship between the digestion, feed efficiency, immune system, gut integrity, and the microbiota management. Yes, intestine is responsible for digestion and absorption, of course, but not only that, but also first protective mechanism against pathogens and toxins. At the same time, additionally, intestine is the largest immunological organ of the body. So for the diseases resistance, it must be well-developed intestine and also the gut integrity. So what are the components of the gut integrity? Of course, the mucus layer is very important for that, to secretion the mucus, and also its protective layer for the against the toxins and the pathogens. And the epithelial cells proliferation is also important for that. And the intestine histomorphological structure is the factors affecting the gut integrity. It must be longer in villi height and lower the crypt depth. And the tight junction proteins is the hard bond protein structure. It's also important for the gut integrity and also the healthy gut. And the microbiota is a big part of the gut integrity. So I will mention about that a little bit. The diversity of bacterial species in the gut is one of the most important factors for the establishment of the stable ecosystem in the intestinal tract. This is suggested by the observation that until the bacterial populations are fully established, young animals have fewer bacterial species in the intestinal flora than adult birds, and making their gut microflora more susceptible to disturbances than that of adult animals. So the age and the fit is the very major component for the microbiota, but the breed, gut region, maternal factors, sex, housing, hygiene, medication, temperature, litter, and location also the other components for the establishment of the microbiota. You can see in this picture some beneficial and the pathogen bacteria in the different parts of the gastrointestinal tract. Most of them the Lactobacillus spp, and also that have the Clostridium and E. coli, but it's not causing a big problem here. So if the gut integrity disruption and of course the performance reducing, so some feed additives can be used to alternative for antibiotics until 2006. That are probiotics, prebiotics, symbiotics, postbiotics, organic acids, and phytobiotics. This feed additives mode of action is for the gut environment and immune organs, changing the gut microbiota and affect the immune-related gene expression positively, and then the enhanced immune response. Yes, yeast is a feed additive. We can use it in all animal rations, all animal diets. I called it the symbiotic because yeast, when we use it alive yeast, it's a probiotic, but when we use the yeast cell wall components, it's an probiotic. So it serves as a prebiotic mainly because it stimulates the growth of beneficial microorganisms in the gut, and they modulate immunity because they are well recognized by the receptors of immune cells. And also they are non-pathogenic agents and support the production and regulation of immunity-related factors such as cytokines. I would like to mention about the cell wall components that are MOS and beta-glucan. These are the strong bonded with them in the cell wall, yeast cell wall. MOS is a prebiotic and beta-glucan is an immunomodulatory effect feed additive. MOS is a probiotic, but a little bit different mode of action other than the other prebiotic. It's not stimulates the growth of beneficial microorganisms. It allows pathogenic bacteria to be expelled without damaging the host by forming strong bonds with the attachment sites of the small intestine. And also MOS alter the gene expression of some receptors and also the ileal genes, for example, interleukin, interferon, something like that. And it's affect some immunological organ weights, such as Bursa fabricius thymus. And beta-glucan, the other component of the cell wall, it's immunomodulatory feed additive. It's affect the macrophage activation and increase the production of cells in the neutrophil. and also they increase the effectiveness of the antibodies. And again, increase the relative rate of the Bursa febris use, and also promote the immunity by stimulating the growth and function of white blood cells. It's not well known that the inulin is another yeast-derived prebiotic. When we use it supplemented in the broiler diets, it's expressed the higher immunoglobulin A and in cecal and mucin mRNA in genome. And this immunoglobulin A are the most important antibodies produced in response to against antigens in the mucous membrane. So yeast cell wall is very, very effective for the gut health and the gut integrity, and also reduce some effect of the diseases, something like that, coccidiosis, Newcastle disease, infectious bursal disease, Salmonella, Clostridium perfringens, and also E. coli. I would like to give some example from our research results here. In this research, we use the different cell wall components. We use the autolyzate yeast. This is the all cell wall, and also the other one is debonded cell wall components. Most and the beta-glucans are debonded, and also the pure most and beta-glucan. In this research, Salmonella infection is very decreased in the experimental groups than the control group, especially in the debonded cell wall components. Again, we can see in this picture the microscope of results of our study. We can see the more cell proliferation, and also the higher villus height in the experimental groups. When we look at the gene expression of the cecal tonsils, the interleukin 1 and 10, we observed that in the this gene expression is higher, is increased in the debonded cell wall components used group. Yes, in the conclusion, health and nutrition are interdependent. The interaction between two occurs largely in the gut, of course. Thus, the term gut health is a very broad topic that requires a multidisciplinary approach involving gut physiology, endocrinology, microbiology, immunology, and of course nutrition. And the yeast has a promising role as a feed additive to replace frequently used antimicrobial growth promoters. These serve as probiotics and prebiotics, postbiotics, symbiotics, something like that, and confer beneficial effect on the performance and the health of broiler. When we talk about the yeast cell wall, have exhibited various noticeable properties like modulation of growth performance, gut microbiome, enhancement of gut development and integrity, immune system, and nutrient absorption and utilization. So the yeast cell wall component is important for gut integrity, and also this means that the healthy gut. So I can say that the gut is the second brain, and thank you for your attention.
Thank you for your lovely presentation. And do we have any questions for Professor Özge? I guess we don't have any questions. Thank you again. And we are going to move on to Mikolaj Adamek's talk. Don't let it under your skin. And he's going to talk about vaccination protects barrier of common carp.
Thank you for inviting me here. I will try to share my screen, which Seems to be not working. Okay. Can you see my presentation?
Not yet. Now, yes, we can see your presentation. Can you,
Okay.
could you move one slide further? Yes, okay, thank you.
Okay, so here we are right now moving to another environment and another universe. I will tell you something about what we're working about in fish diseases in our unit. This is only one very small cutout of our work, because we work with every pathogen of fish and also with fish welfare. But here we will be focusing on a virus and viral infection. First, we have to put ourselves in the shoes of our animal. So this is a grumpy fish, and one would probably ask why this fish is grumpy, because it is a freshwater fish which is somehow encapsulated in a very hostile environment, which is the water. The water has a wrong osmolarity and has a lot of secondary pathogens. And fish developed over the evolution some mechanisms to protect themselves. And one of these mechanisms are the mucosal tissues, which are also including skin and gills of fish. And they are protecting them from this hostile environment. However, when epitheliotrophic pathogens occur or happen to encounter the fish, there might be some problems with the this barrier, and we would like to study this based on koi herpes virus disease, which is a mucosal disease. This disease is global, and normally it ends up with a lot of dead fish, which are having a quite drastic clinical signs on their gills and skin. And we decided that we will try to first vaccinate against this disease, and secondly, we will look how these vaccines work and how they are protecting fish from the primary pathogen and how they are supporting the barrier of fish, mucosal barrier of fish, to protect from secondary pathogens. So we decided to use one of our colleagues' vaccine candidates, which was a recombinant live attenuated virus, and we focus on how this vaccine is inducing immune responses, how it is affecting osmotic balance, skin integrity, and secondary infections. And we did this both during the primary infection with the vaccine and also during the experimental challenge with a wild-type hypervirulent Cyprian herpes virus 3. So we decided to do a quite complicated infection scheme. We will focus on these two groups, which will be presented with these two pictograms. The vaccinated fish have the shield, the non-vaccinated fish have no shield, and we will follow these two groups over our experiments. So first, we did a vaccination, which is an infection with this live attenuated virus. There was no mortality. However, the virus established itself in the fish and was growing over the time, while till, let's say, 7 days post-infection, and then it was starting to be cleared from the fish. We have several markers for looking if the disease is occurring. One of them is sodium level in blood. And the sodium level in blood didn't change. So also the bacteria flora, which here is presented with a full bacterial load on the skin, didn't make too much of changes. There were some increases in the vaccinated fish and also decreases in some of our adjuvants, but these differences were very minor. So we can conclude that our vaccination didn't cause the outbreak of the disease, which is good. We looked at the immune responses, and I'm presenting only a very small fragment of this. We had some IgM responses, and we had also thermoneutralization titer increasing in the blood over the time of vaccination. So then we decided that we will challenge this fish with the hypervirulent virus, and here the outcome of the infection is quite drastically different for both of these groups. Our non-vaccinated fish unfortunately had to be removed from the infection because they developed a very severe immune response, very severe pathological changes, and the mortality was starting very drastically after 5 days post-infection, while our vaccinated fish didn't budge. They were swimming happily in water and being not affected by the virus. We look at the virus load, and we see as drastic differences in virus load, especially at day 7. So this was when the infection was really in full blown, and we see that vaccination was limiting the virus load in skin. So we have a good model. The vaccine works. So now we could look why it works and how it works. First, we looked at the skin responses, immune responses. Skin is the main portal of entry of the virus, and we have a situation of good news, bad news. The good news is for vaccinated fish. They have several vital immune responses upregulated, which are most likely protecting them. So antiviral response is boosted also in the vaccinated fish, while there is very low virus load. The neutrophil responses are boosted, the anti-inflammatory responses are up, and B-cell responses are up. While in the non-vaccinated challenge fish, there are only bad news. The bad news, the first bad news was that there was some hallmarks of immunosuppression with T-cell responses, which were down in skin. Inflammatory responses, pro-inflammatory responses were up, and also some indicator of stress were up. When we look closer on the skin, we see a very drastic pathological changes. So we have a loss of scales in the fish, which was not vaccinated, and it can be up to 60% of the skin surface is losing the scales, while vaccinated fish have no problems with scale loss. The scales are intact. When we look in histology, we see in vaccinated fish a normal skin histology with a scale here at the bottom, covered with the epithelium, with goblet cells and the mucus on top, while in non-vaccinated fish, we see that the fish lost the epithelium over the scale or lost the scales with the epithelium entirely. When we look at gills, which is the second target of the virus, we see some changes. We see increase of the thickness of the secondary lamella, while in non-vaccinated fish this is not occurring. And what is important is that we see the drop of the sodium concentration in non-vaccinated fish. So they are developing full-blown koi herpes virus disease. And these sodium levels are quite lethal for the fish, while the vaccinated fish have no problems with sodium levels. When we look closer, and I will go really fast through this, on the skin barrier on the molecular level, what we see is that several very important parts of the mucosal barrier, including mucus, adherent junction, desmosomes, and tight junctions, are disrupted in the non-vaccinated challenge fish, while vaccinated fish have no changes in this skin barrier molecules. So what would be the outcome of the disrupted barrier of fish? Fish are swimming in water, and they are surrounded by facultative bacterial pathogens. And we looked at the bacterial load in the skin And here you can see the differences in universal bacteria primers, which show that there is a huge increase of bacterial load in the non-vaccinated fish, while the vaccinated fish keep their bacteria flora on a quite low level. And here we have a couple of other bacterial species which we measured, and we see the same effect. So bacteria flora is disrupted and it is increasing, and it is also increasing in the content of the pathogenic bacteria, which are breaching the skin barrier and increasing the pathogenicity or pathological changes during the disease. To summarize this, we have a live attenuated vaccine which works by increasing the neutrophil T and B cell responses, and this provides the protection from the primary pathogen, but also preserves the gill and skin integrity and protects from osmotic disbalance and additional bacterial pathogens. So the take-home message would be that vaccination against cyprian herpes virus 3 works in several ways, protecting the fish from the primary and secondary pathogens and also environment during the life of the fish. So this kind of research is not done alone. I would like to thank the collaborators and also the funding agency, and I'm looking forward to collaborate with Ankara.
Thank you very much for your presentation. Do we have any questions for Dr. Mikolaj? I guess we don't. And we can continue with Dr. Farah Gonul Aydın's presentation. Gonul?
Hi. Yes.
Hi.
Just a second. Sorry. I think it's okay.
It's
you
okay.
Can see.
Yes, you can see. Yeah.
Okay, then I can start. Hi everyone. First of all, thank you for the opportunity and previous speakers. I'll give you brief information about antibiotic drug resistance in fish pathogens and look into the AMR under One Health umbrella. But first, before started, I'd like to introduce myself a little bit. I am a postdoc researcher at Ankara University Pharmacology and Toxicology Department and certified aquatic veterinarian, and more than a decade I am studying just aquatic animal health. Given the time, I will try to give you a brief overview about aquatic animal production, then continue fish health and treatments, AMR problems, and One Health approach, and I will close with international strategies addressed to AMR, not just aquatic, but also terrestrial animals. As we all know, aquaculture has been practiced for thousands of years. An early form consists of trapping with wild aquatic animals in lagoons, ponds, or small lakes. Nowadays, it's estimated that more than 600 aquatic species are cultured worldwide for production in a variety of farming systems using freshwater, brackish water, and marine seawater. Aquatic food products represent one of the world's most nutritious and healthy food sources and is the major contributor to food supply at global level. It is at present one of the fastest growing food producing sector in the world. In EU, European sea bass and jilted sea bream are the main fish species farmed in the Mediterranean region. They are produced in land-based extensive and intensive grow-out system and the sea cages. European sea bass and the jilted sea bream are farmed almost entirely in 19 Mediterranean countries, although more than 90% is concentrated in Turkey, Greece, Egypt, Spain, and Tunisia. Total juvenile production is concentrated in 5 countries. More than 90% of fingerlings are produced in Turkey and Greece. When you look at the slides, both sea bass and sea bream and trout production getting increases since the global population and food demand raises. A high number of factors through the farming period may thus affect fish welfare, including the physical and chemical environments, feeding, social interactions, occurrence of fish pathogens, parasites, and predators, and the procedures during handling, transporting, and slaughtering. Despite the technological process applied in recent years to the various productions, from reproduction to nutrition, several welfare and health issues of farm fish still represent relevant limiting factors for sustainable production. In particular, the attempts to increase and diversify production have been often accompanied by the emergence and reoccurrence of transmissible diseases, strongly conditioned by management and environment factors, with consequently significant economic losses and repercussion on fish welfare and health parameters. Total losses related to fish health impact of the disease around 20% of European aquaculture value per year. Main fish disease can be classified as transmissible diseases and non-infectious diseases, but multiple pathogens affect farm fish, often with co-infections. And although these are the main reasons, don't forget the parasites and pathogens are the parts of the natural ecosystem for aquatic animals. As you can see, the whole slides can be seen diseases for fish health. Aquatic animals living in the wild, as well as the rare aquaculture facilities for consumptions or ornamental purposes, are susceptible to a diverse collections of bacterial pathogens. Aeromonas species, Vibrio, Streptococcus, and Adversia latarda are the main pathogens for causing diseases in fish. Thus, the use of antimicrobial drugs in the treatment and prevention of diseases was chosen as a method to solve this problem. The use of these drugs in aquaculture is important for successfully treating sick fish and maintaining the health and well-being of the animals. Very rarely it is possible to control bacterial diseases in aquaculture by eradication. Well-targeted antimicrobial chemotherapy remains vitally important for the treatment of bacterial disease. As you see, the most common antimicrobial drug classes used in farming dominant aquaculture species. Tetracycline and the penicillins tetracyclines and phenicols are the most used class in aquatic environment. 15 major producing countries, about 70% apply to oxytetracycline, sulfadiazine, and fluorphenicol, while 55% use sulfamethoxazine, erythromycin, amoxicillin, and enrofloxacin. Global microbial consumption, Global consumption in aquaculture 2017 was estimated at nearly 11,000 tons. As you see, aquatic medicines is different from terrestrial animals because the dosage related to centigrade temperature and their routes, more affecting our antimicrobial treatments and protocols. In aquaculture, we use antimicrobials for 2 reasons: benefits of the use and concern related to these concerns. Benefits of the antimicrobials, of course, for the culturing methods to prevent our failure for the therapy, emerging and re-emerging diseases, infectious diseases, and developing cultural technologies. Antimicrobial help us for getting through these problems. Yet, when we use or misuse these antimicrobials, can be difficult or cause more problems. Generally, bacterial diseases are controlled by feeding infected fish with antibiotic-medicated feed based on sensitivity testing. The efficacy of treatment via medicated feed depends on the rapid diagnosis and commencement of treatment, selections of appropriate microbials, proper dosage, and durations of the treatment. However, this practice may be ineffective because sick fish lose appetite. In addition, frequent use of antimicrobial compounds has led to the development of resistance to antimicrobial compounds in pathogens, posing serious challenges to both aquatic animal health and human health. Antibiotic resistance bacterial cause over 35,000 human deaths annually in the United States, where 33,000 in European Economic Area. In India, it's up to 58,000 and probably more in Southeastern Asia. And these numbers are expected to rise due to rapid socio-economic development and population growth. Although the precise quantities of antimicrobials used in aquaculture are mostly unknown, especially in low middle-income countries. Although AMR is observed in aquatic bacteria, it's difficult to find a direct link between the resistance profile and antimicrobial usage. Therefore, there is a need to be cautious while drawing the conclusions about the source of antibiotic resistance in bacteria associated with aquaculture. Again, we compare to the human and terrestrial animals, numbers are not quite enough for determine the resistance profile in aquatic animals. AMR in pathogens of aquatic animals has been reported from different systems. Over 80% of Vibrio harvae from finfish aquaculture system in Italy showed resistance to amoxicillin, ampicillin, and erythromycin, while 76% of strain Vibrio show resistance to sulfadiazine, and most Vibrio vulnificus strains isolated to Dutch eel farms showed resistance to cefoxitin. Even though this antibiotic was not used in eel aquaculture, can be seen their effects. Also, positive correlation found between high concentration of tetracycline or fluoroquinolones in certain water samples and resistance to these antibiotics in Aeromonas and Vibrio species. As you can see, when we look at the epidemiology of antimicrobial resistance, it's very various and it's very complex and it's integrated all the systems. Antibiotic resistances may have evolved naturally. Indiscriminate use of antimicrobials in humans and animal sectors has led to the selection and spread of the resistance bacteria. RGs found in aquatic system are derived multiple sources. Animal farm and hospital effluents reach the aquatic environment. Antibiotic resistance determinants selected to other environments can also be found in aquatic environment and then may reach products of aquatic animals. Currently, the molecular mechanisms and prevalence of quinolone, tetracycline, and penicillin resistance genes represent a great challenge to control diseases in aquaculture environments. Antimicrobial resistance genes present in pathogens associated to major global diseases reported in aquaculture. And you can see the drugs, diseases, and their resistance genes and how they seen each part. Antimicrobial resistance determinants found fish pathogens and other marine and freshwater bacteria share with human pathogens also. In this slide, you can see the same resistance genes found in human, and it causes the same resistance to antimicrobials for every human. AMR is known to have multiple drivers and needs to be tackled on many fronts. Hence, a One Health approach is essential to ensure that all sectors and stakeholders communicate and work together effectively. One Health is a collaborative multisectoral and transdisciplinary approach that recognize and intersections between people, animals, plants, and their shared environment. The European Union Commission recognized the importance and awareness of AMR in 2001, and after that, the Community Strategy Against Antimicrobial Resistance was set up, and first policy instrument addressing AMR at European level published. In 2011, the policy was reinforced with the action plans against the rising threats from AMR using the One Health approach, covering both the human and animal health sector. Global efforts include the 2019 United Nations Political Declaration on AMR2 and 2015 AMR3, and which was subsequently adopted by World Animal Health Organization, OIA, and Food and Agriculture Organization. AMR has also been addressed lately G7 and G20 forums. As a consequence of One Health, AMR monitoring in animals should include not only food producing animals, but also pet animals and other animals like ornamental fish. The strategies regarding AMR aquaculture considered in the guidelines for the prudent use of antimicrobials in veterinary medicine, and principles is that similar strategies used for reducing the AMU in terrestrial farm animals should be also used in aquaculture. EU level supporting One Health, national antibiotic programs, joint actions, increasing assistance of EU Health Security Committee and Commission's working group on AMR in veterinary and food safety area. Being a working group, our missions establish antimicrobial treatment guidelines to help national organizations to adopt these guidelines on their national levels. For global response addressing AMR, we can talk about, or we can see resources, material, global action plans, and three parties joint declaration published in their specific websites, antimicrobial resistance, fighting antibiotic resistance. OIA standards and guidelines related to AMR, both aquatic animals and terrestrial animals, published recently, early 2021. And last month, also WHO published resource materials for in-country development and implementation of national action plans to address microbial resistance. And again, three parties published for international instruments on the use of antimicrobials across the human, animal, and plant sectors. AMA, also European Union, also published their own regulation. And to prevent resistance, we could keep going to fight in every level. I like to finish my presentation with a quote. Antibiotics are like fire extinguisher. We don't want to use them all the time, but we are happy we have them on the wall. So lastly, this year's World Antibiotic Awareness Week 2021 is getting closer, and this program has a topic, and it's spread awareness and stop resistance. And if we have all played a role, and you as a part of the antimicrobial users can help to prevent these fight. And thank you for listening to me and my team. Lastly, spreading awareness because we need you. If you have any questions, I can take you.
Thank you very much for your presentations. Do we have any questions for Gönül?
I think no one wants to know. Actually, next week we also have in antibiotic resistance week. We also have a big program to contribute fighting this antimicrobial resistance, both terrestrial and aquatic animals. And if anyone want to include, they can reach me.
Okay, you can share your email in the chat room.
Okay.
So since we don't have any questions, I'm ending the session here. And we can be back at 3 in Turkey time and 1 in German time. We are going to give 8 minutes of break.
Thank you.
So see you in 8 minutes.
Thank you.